Ladies and gentlemen, good morning. It's early morning where I am. It's about what's the time? 2:35. Just a perfect time to be looking at anatomy and physiology. So you are coming from a discussion of skeletal muscle. So here we are just trying to highlight that there are a few differences with smooth muscle. For cardiac and skeletal muscle, the contraction is almost the same. For smooth muscle, it's slightly different and you need to pay attention. So in terms of the histology, I think we've already discussed and you can actually see the shape of the cells. You also need to remember that they are unicellular. You also need to remember that they are autonomically controlled. You also need to remember their contraction is modulated through the endocrine manner. You also need to remember that they are found in internal organs, blood vessels, GI, urogenital organs, walls of those organs, and the dermis of the skin. So again, here we are just showing the histology, the shape of the cells. Now here is where we are highlighting the issues concerning smooth muscle contraction and the slight major differences between skeletal muscle, cardiac, and smooth muscle. So for smooth muscle, while in the skeletal muscle, the calcium binds to troponin, here there is another molecule that you're going to find, calmodulin. So calmodulin is the control or regulatory protein for smooth muscle instead of the calcitonin, instead of the troponin and tropomyosin. So calcium released after contraction, after depolarization of the smooth muscle, will be released from the caveolae. The caveolae is like the smooth endoplasmic reticulum, and it is not so very organized, so it's like a food vacuum, and it will bind with the complex of calmodulin. So calmodulin plus calcium, it will make the calcium calmodulin complex. Okay. Now, the calcium calmodulin complex binds to the myosin light chain kinase (MLCK), which becomes active, such that this now calmodulin myosin light chain kinase, which is active, now causes phosphorylation of the myosin. The myosin that is not phosphorylated, it is in a wounded structure, as in winding. Now, when it is activated by phosphorylation, it will begin to unfold and wind, so that now the binding um, uh, components of the myosin can be free now to bind to the actin. Now, the kind of binding you find in smooth muscle, it is described as a latching mechanism. Latching is like you have a baby that is going to breastfeed. The way they breastfeed, they don't just kiss the nipple or the teeth. They actually put the nipple inside the mouth. That is latching. Okay. So even in the separation of the myosin from the actin, it slightly takes longer than the separation of the binding of myosin to the actin in skeletal muscle. So the contractions here are longer and much slightly stronger than the skeletal muscle, and so this has to be taken into consideration. So this is very, very important. The other components that you need to remember is that in the skeletal muscle and the cardiac muscle, there is the sarcomeres. In the smooth muscle, the sarcomeres are not so very organized in such a way as to explain the way we have highlighted in that histology. But here, the organization is such that it's a similar, a fake copy of, it's a pseudo-sarcomere, where the Z lines are exemplified or illustrated by uh, Denise bodies. The Denise bodies, these are like Z lines where the actins are connected, such that when the myosin binds to the actin, they begin to move closer to the Denise bodies, and this is what is called the contraction of smooth muscle. So you need to pay attention to that. And the next pictures are just showing you an illustration of histological appearance of a relaxed smooth muscle versus a contracted muscle. And here we are just showing the differences as a summary between the skeletal muscle histological appearance, the cardiac muscle, and the smooth muscle, and some highlights of the physiology of their contractions. Otherwise, this is the end to muscle contraction. Best wishes in your exam. Bye-bye.